Hello. Sorry for being a little late. Zoom would not let me in. I had to verify who I was before I could even log into Zoom. So sorry about that. I'm going to have to ask them why we don't have a link to Zoom in Canvas. Any questions before we begin? I don't want that. All right, could somebody confirm that you can hear me?
Yeah, I can hear you.
All right, and the volume's good?
Yeah, not too loud.
Good. This time, at least I remembered my password and how to log in. But like I said, it wouldn't let me log in. I had to call them and send a code to verify, I guess, that I was John Volpe. Or somebody else had my phone, they could do the same. All right, today is July 18th, and we are supposed to be starting chapter 2 today. I'm not sure we're there. I think we're a little behind schedule. Please let me note that this Wednesday you have a practice quiz 00. And that quiz covers chapter 1 and the syllabus. It may also cover the lab 00. Let me check on that. You're not seeing my screen, right?
No.
No, okay, let me check on that quiz for you. Of course, I'm not in Canvas because I can't get to Zoom from there, so I've got to log into Canvas now. And I see I've got some emails. I've not checked my emails today. Let's see, where do I want to go? Quizzes. Let's just go down to week 2. Let me look at the questions quickly to make sure that it has chapter 1 and lab 00. Okay, yeah, I'm correct. So mostly chapter 1, a little bit of the syllabus, and lab 00. And I should really write that on my schedule here. Let me start sharing my screen. Let me state that I have done some grading. I know I got lab 00 graded. I may have gotten some other things graded. I have not graded lab 01, meaning lab 1. I was hoping to get that graded, but I ran out of time. Let me share my screen. All right, you should be seeing my screen now. And let me show you the syllabus again. Oh, come on. So we should be starting chapter 2. We're not, we're a little behind schedule. And then there's practice quiz 00. We will have a lab today, lab 2, aseptic transfers. Any questions about what we're doing? All right. If not, let me go to lecture 1. For the practice quiz, you'd need to download Respondus Lockdown Browser, so make sure you do that before you attempt to do the lab or the quiz. And you should try to do the practice quiz because it'll be a good practice for a real quiz, which is worth more points, but it'll be a good practice and give you an idea of what my questions will be like, as well as it'll make sure you have Respondus Lockdown Browser up and running on your system. If you do not, and you have problems, you'll need to contact the computer help desk to get your settings adjusted. And if you do not get Respondus Lockdown Browser running, within 2 days of the quiz, the answers will show, and you may not take that quiz any later. Okay, under no circumstances do I allow students to take the quiz after the answers have shown. So try and get the lockdown browser up and running on your system. And if you take the practice quiz 00, you'll make sure that you do. And then if it doesn't run on your system, it's no problem to me when you take the practice quiz. You can take that one late. All right, so let's talk a little bit about the history of microbiology. The history of microbiology largely concerned two ideas. It seems to me that we talked about this. Did we talk about this? Anyone?
I don't think so.
All right, let me just.
We
I don't think we did. I think we may have
think-
mentioned a little bit about spontaneous generation, but not fully into
yeah,
it.
like
All right,
like
yeah, I
just
don't have
started.
another mark. Okay, so the history of microbiology concerns two competing hypotheses to explain how life began. One, spontaneous generation. Life arises from inorganic matter when the matter is mixed, and then a vital force comes in and forms life. They never explained what the vital force was, but it's some force coming from outside and creating life. And then the other theory is biogenesis, that all life arises from pre-existing life. And there never was a question about where human babies came from. It was never spontaneous generation, but there were arguments put forward that life could spontaneously generate. There were beliefs over a century ago that included things like you threw a log into the fire and a newt would arise from it. And people would say, spontaneous generation. The wood and the fire with the vital force coming in creates the newt. And then people saw flies emerging from manure. They thought that was an example of spontaneous generation. And then maggots arising from a decaying corpse. Most were logical, none were scientific, and some were amusing. Like somebody wrote up an article, I think it was actually a scientist too, that if you put sweaty underwear in a jar that had wheat in the bottom of the jar, it would spontaneously give rise to mice. In reality, the mice got into the jar to eat the wheat, and then because it was in a jar, the mice couldn't climb out of the jar, and the sweaty underwear actually had nothing to do with the mouse being in the jar. A little sidetrack, and you could tell that there's growth in the tube by looking at the tube. Like here is a tube of media, and you can see the media is clear. When there are microbes growing in the media, it will be turbid or cloudy like this. Any questions about any of that? And this was another reason why they suspected that especially microbes could generate spontaneously. Whenever they poured media into a container, later, when they observed it, it was cloudy. And it was a long time before people came to the conclusion that microbes could fall from the air, and then that microbes were already in the tube. The air one was a little bit longer in coming, meaning that they did sterilize the media and sterilize the tube, but they were a little slower at realizing that microbes could fall out of the air. And actually, Louis Pasteur is the one who proved that. The first evidence, either for or against spontaneous generation, came from Francesco Redi. And you should actually know this man because he's also the first scientist who wrote up a controlled scientific experiment. You don't need to know when he did this in 1668. Francesco Redi actually got 3 jars that he completely sealed, and he put meat inside them before he sealed the jar. And he had 3 open jars, and he discovered maggots appeared in the meat in the open jars, but not in the jars that were completely sealed. And the spontaneous generation people said, well, that's not going to work. You've sealed the jar. You've kept out the vital force. So that's why there's no maggots in the jars that are sealed. And then Francesco Redi did something really brilliant. He took the jar, put the meat in it, and he had 3 jars like this, and then he covered them with a fine wire mesh net so that the vital force could get in, but other things could not. And he saw no maggots in the meat. Later, it was learned that maggots were getting in the meat because flies were coming into the meat that was in the open jar, laying eggs, which gave rise to the maggots. But that was not known in Francesco Redi's day. That was learned after he did this experiment. But anyways, this is the first controlled scientific experiment, and with it, he showed no evidence for spontaneous generation. And after his day, meaning after this experiment and his publication of it, scientists generally agreed that large organisms had to come from another pre-existing organism. Okay, meaning the maggots came from the flies and babies came from the 2 adult parents. However, people argued that microbes were smaller and simpler and that they could spontaneously generate. And Francesco Redi's experiment had no observation on microbes. I think in Francesco Redi's day, microbes weren't even known, or if they were known, it was just when they were starting to be known, meaning that this was before really microbiology started. Yeah, the first observation of microbes happened really by Antony van Leeuwenhoek, and I'm not Dutch, so I could be butchering his name, although I think Antony is right. It's the van Leeuwenhoek I'm not sure of. He observed the first microbes, and he described them and wrote about them and published them. And wherever he looked, he found microbes. He did scrapings from his teeth. He looked in rainwater. He looked in peppercorn infusions. And then he looked at the water from the River Thames, which was where London got its water supply. And he found microbes in all of these areas. And people weren't too happy that the water supply of London had a bunch of microbes in it. Francesco Redi was the best microscope builder in his day, and he made the microscopes himself, and he also was the best lens grinder in his day. So he actually painstakingly ground down the glass to form the lenses. And you'll notice he only made a simple microscope, okay? But his simple microscopes could magnify much better than the compound microscopes that were being made in his day. Having only one lens, it was easier to focus on a specimen, and so he got much better clarity and resolution than anyone who had a compound microscope, which the compound microscopes could magnify further than his, but everything was blurry. And so you could not see microbes in his day. He saw cells that were so small they were not seen for another century. He saw these cells here, which if you look at them, that looks like a rod, a bacteria. That's another rod, and he's showing the movement of it. That looks like a spirochete. Figure E is showing cocci, two cocci, and then figure F is showing various rods. So he was the first person who saw and actually drew examples of bacteria. Okay, so when we look at this, we know these are bacteria. Bacteria were cells so small that nobody else saw them for 100 years after he saw them. And after 100 years, what happened was the compound microscopes got better and better. And after 100 years, they finally got good enough to actually see bacteria. Any question about any of that? All right, so Antony van Leeuwenhoek described the first microorganisms, and he was the best lens grinder and microscope builder in his time. He was actually only an amateur, biologist or microbiologist. He never did it for full time. How he actually made his money was he was, a tailor, somebody who sold cloth. And he made his microscopes initially to observe the cloth because he thought if you looked at it under high magnification, you could learn better things about it. But like I said, he did look at living things, including microbes. Okay, after Antony van Leeuwenhoek reported seeing microbes wherever he looked, Rudolf Virchow proposed an extension of the cell theory, stating that all living cells arise from pre-existing living cells. So he wrote it up, although the theory of biogenesis was known before 1858 when Rudolf Virchow wrote it up. And this concept became known as the theory of biogenesis, which stated that all living things come from a pre-existing living thing, including microbes. And the argument about whether spontaneous generation of microbes occurred or biogenesis of microbes occurred continued until 1861. Who solved this debate was Louis Pasteur. Louis Pasteur disproved spontaneous generation and therefore confirmed the hypothesis of biogenesis when he demonstrated first that microbes are present in the air and they could fall out of the air and contaminate something that was previously sterile. He also showed that microbes could be destroyed by heat. He then further went on to show that spontaneous generation of microbes did not occur with his famous and ingenious swan neck flask experiments. What he did was he got a flask with a long neck and he poured media in it, And then he heated the neck of the flask, bending it into, they call that a swan neck, where it's sort of like an S shape. And then he heated the media, which would sterilize the glass of the container, as well as the media. And then he let it cool. And he noticed that if he let it sit this way, that there would be no microbial growth in the flask. In fact, if you ever go by the microbiology lab, in the window of it, there is a flask there that has been there for over 10 years. And somebody repeated Louis Pasteur's experiment. And this has been sitting for over 10 years, and maybe over 15 years now. But it's been sitting in the window of the microbiology lab. Eventually, the media is going to dry out, and I'm not sure what they'll do. They'll probably throw it away at that point. He did show that you could get microbes growing in here if you took the media and you brought it down to here and then brought the media back into the flask. What's happening is the microbes from the air would fall into the tube. And I should say this tube is open to the outside to let the vital force in. And what happened was the microbes from the air would fall into the tube, but then they would stay here. They would not go up the tube against gravity unless you actually brought the media down into here and then brought it back. This disproved the theory of spontaneous generation of even microbes. And Francesco Redi had already disproved it for larger complex organisms. Any question about Louis Pasteur's experiment? All right. With Louis Pasteur's work, it began the discovery of many things, like the relationship between microbes and disease, giving rise to the germ theory of disease. Out of that work came immunity, that our bodies could respond to microbes with an immune system, and that's known as immunity. And then out of that work also came the concept of antimicrobial drugs, that we could use drugs to prevent the growth of the germs, the microbes that cause disease. Any question about any of that? Pasteur also developed the technique of aseptic technique, which was important in microbiology because when you wanted to study a single species, you would isolate it, only have that in the culture, and then you had to use aseptic technique to keep it sterile. Because if a culture has more than one species in it, and then the test is positive, you do not know which species in the culture caused the test to be positive. Is it both of them, or is it one or the other? And there's just no way to know unless you're working with a pure culture. And Louis Pasteur did come up with a technique to get a pure culture. We'll talk about that later in the lab, I think. His technique was not very easy. I guess I can talk about that now. And that is what he did was he would make dilutions of a sample containing a microbe, and he would make a dilution of that and several dilutions, meaning different concentrations of microbes in the dilution. And then he would have a replica for each dilution. So he put that dilution into a tube, and then he'd have 9 other tubes of that dilution. And if he found in one dilution that only one tube had growth in it, he correctly concluded that probably one or a few cells caused the growth in that one tube. And so that tube was likely to be a pure culture. And then the other 9 tubes there's no growth in it because instead of one or a few cells being added, it was no cells were added. And he would use that tube to set up his pure cultures. Any question about that? Anyways, that was a very complicated way of getting a pure culture. You had to make a dilution and then several dilutions of that. And then you plated each of those dilutions and you had to make a replica for each dilution so that there were 10 tubes of each dilution. And then you had to look for one which only had one of the 9 tubes showing growth. 9 of the tubes had no growth. And there's all sorts of problems in this. You could have one tube with 2 tubes showing growth, and then the next dilution having no tubes showing growth, in which case he'd have to start all over again. And if he didn't dilute it enough, he would have his most dilute dilution having more than 1 tube showing growth. Robert Koch developed a much easier technique for getting a peer culture, and it involved streaking for isolated colonies on a solid media. If you streak your bacteria on a solid media, then 70% of the colonies will be pure. And that isn't quite good enough in microbiology, but if you take an isolated colony and you plate it on a second culture and then streak it for isolated colonies, so that the cells have gone through 2 colony formation events, you increase the chance that it is a pure culture to over 95% of the time. And that is what microbiologists do when they're trying to obtain a pure culture. They streak the cells out 2 times, meaning you streak it out for isolated colonies 1 time, and then take an isolated colony, and then you streak it out on a new plate for isolated colonies. And then you harvest from 1 isolated colony where the cells have gone through 2 colony formation events. Any question about any of that? All right, about the same time, a technician who was working in Dr. Koch's lab, developed nutrient agar, a very good general purpose media that was actually cheap to make and use. That's why we use it a lot in microbiology, because it's cheap, as well as the fact that it grows many different organisms. Many things can grow on nutrient agar. About the only thing that cannot are fastidious organisms. And a fastidious organism is one that requires specific nutritional additives before it will grow. Any question about any of that? All right, about this same time was when Sergei Winogradsky developed the idea that microorganisms were involved in the biogeochemical cycles on the planet Earth. And we talked about the nitrogen cycle and how microbes largely transfer nitrogen from one place on the planet to another place, and then they change the form of that nitrogen. And then nitrogen recycles on the planet, moving from one form in one place to another. Well, this also happens with the sulfur cycle and the carbon cycle, and Winogradsky developed this concept. The reason why Winogradsky was able to develop this concept was, unlike other microbiologists, Winogradsky studied mixed cultures of microbes. And to see the nitrogen cycle, you have to have all of them in there that can recycle the nitrogen. So you do need many different species to recycle the nitrogen. And Winogradsky just sampled from the environment, and he studied many different microbes living in a community, meaning living together. Any question about any of that? All right. Pasteur also showed that microbes were responsible for fermentation. Alcoholic fermentation is a conversion of sugar to alcohol to make beer and wine. And the beer industry actually asked Louis Pasteur to help them figure out why beer sometimes spoiled. And when it spoiled, it was sour and not very tasty and not drinkable. And Louis Pasteur discovered that what was happening is the microbes were fermenting the starting ingredient, which was grape juice for wine. And I'm trying to remember what the heck the starting ingredient is for beer. I'm losing it. So I think it's hops and grain, but I'm not real sure because I'm not a beer drinker and I've never made beer. But he showed that the microbes were responsible for fermenting the starting ingredient to make the beer and the wine, meaning make the alcohol. But there were other microbes that were responsible for changing the alcohol into vinegar. And that was by the production of acetic acid by other microbes, fermenting the alcohol to acetic acid and then spoiling the beer or the wine. Excuse me. Louis Pasteur then went on to find a problem for preventing the beer from spoiling, and he invented pasteurization, where the spoilage bacteria could be heated and killed, although it was not hot enough or long enough to evaporate all of the alcohol in the beer or the wine, meaning you would evaporate some because you didn't need to heat it, but most of it would remain because the heating was not that hot and not that long. It did not make the beer or the wine sterile. All it did was remove the spoilage organisms. There were microbes in the beer or the wine. And you're probably familiar with pasteurization, the application of a high heat for a short period of time. It's used in a wide variety of foods, but Louis Pasteur invented it to pasteurize beer so that the beer would not spoil. And it does reduce the number of microbes in the beer, as well as when you pasteurize milk, it reduces the number of microbes in the milk, but it does not remove them to zero, so that pasteurized items do have a shelf life. And after that point, they can spoil. And I think everybody knows with pasteurized milk, it does last longer than unpasteurized milk, although you probably don't know that, but you can keep milk in the refrigerator unopened for the expiration date, which is something like 2 or 3 weeks from when it was first made available in the store. And then it can be used, for a couple of days after that, but it should be sold by that date.
Yeah, I buy raw milk at the store, and that stuff expires in like 7 days, man. It sucks.
Yeah, raw milk. How can you buy raw milk in the store? I thought it was illegal in all the states of the United States.
Oh, you can buy it at Chuck's.
In what?
You can buy it at Chuck's Produce.
Okay. I'm surprised you can buy it because I thought it was illegal. Maybe it's just the state that I used to be from, Oregon, which did not allow unpasteurized milk to be sold. But I thought in the United States you could not buy raw milk because of the risk of getting an agent that could kill you. Now today, maybe they have other ways of removing the microbes from the milk, but pasteurization is the main way, and it removes only the milk spoilage organism as well as the organisms that cause human disease. It does not remove all organisms. And that's why pasteurized milk has a shelf life. But as was stated by the student, the shelf life is longer than raw milk. And most of you probably wouldn't know that, but it does last more than a week, and almost nothing in the refrigerator lasts longer than a week. And when you open the pasteurized milk, microbes from the air can fall in, and so you should use it within a week of when you open it, even if the expiration date is after that time. Meaning once you open it, you should only keep things in the refrigerator for about a week unless it has preservatives in it. And we'll talk a little bit more about that later. So with the concept of fermentation, the realization that microbes can affect organic matter and then change grape juice into beer and wine. The question arose, could microbes have similar relationships with plants and animals? And may microorganisms cause disease? This concept became known as the germ theory of disease. Originally, poisonous vapors were believed to be responsible for at least some infectious diseases. They didn't have the germ theory of disease, for one thing, earlier, because they didn't even know about germs, but the concept that microbes could cause disease became known as the germ theory of disease. And before that, people believed you breathed in poisonous vapors. So when somebody went walking through the swamp and they got cholera, they believed it was from the person breathing in the poisonous vapors of the swamp. It was not that the person was exposed to cholera that happened to be in the swamp, and then they breathed in the cholera, which is really what happens, by the way. We'll skip down. Robert Koch was the first person to actually prove the germ theory of disease. What he did was he showed that the bacterium, Bacillus anthracis, causes the disease anthrax. And then he wrote up his steps that he used to show that Bacillus anthracis causes the disease anthrax. And these specific steps that microbiologists use to show that a given organism causes a given disease is known as Koch's postulates. The steps we use to show that a given microbe causes a given disease. With this, Dr. Koch actually proved the germ theory of disease. He did not, let me state this in very clear terms, he proved the germ theory of disease. He did not propose the germ theory of disease. Okay, whoever was the first person to bring up the concept that germs may cause disease, it was not Dr. Koch. And to tell you the truth, so many people proposed it before it was proven. I'm not sure we know who the first person is who first proposed it, although somebody may have found the first written proposal of the germ theory of disease. But it was a concept that many people believed before Dr. Koch showed it. And let me state, this will be the first of 3 times we're going to talk about Koch's postulates. So this is an important topic, and we will return to it 2 additional times. A little before Dr. Koch's time, was the first vaccination, where Edward Jenner, you don't need to know the year, but in 1796, he came up with the first vaccine protecting against smallpox. What he did was he inoculated a person with the cowpox virus. And that person was then protected against smallpox. Nobody understood why Edward Jenner's procedure worked, or even why cowpox was protecting against smallpox, until Louis Pasteur once again showed what was going on in vaccine. And what he did was he showed that he didn't do it with smallpox and cowpox. He actually did it with his work on a vaccine causing a disease in chickens. So it was a viral disease causing disease in chickens. And anyways, what he did was he showed that the vaccine, if it was similar enough to the organism that we were trying to prevent, like cowpox was genetically similar to smallpox, when the immune system responds to attack cowpox, some of it will also go on to attack smallpox. And that is how the Edward Jenner vaccine, meaning cowpox, protected an individual from getting smallpox. It's just that the viruses were very similar to each other, meaning they had a shared evolutionary history. And then when the immune system responds to one, it responds to both of them. And after this work, people were able to make further vaccines. When they didn't know how the vaccine worked, nobody knew how to make a vaccine. But after Louis Pasteur showed how vaccines work, people then began making different vaccines. And Louis Pasteur himself made several vaccines. The first was some chicken disease. He made a vaccine against that to help the chicken industry. He also is famous for making the rabies vaccine. He was the first person to come up with the rabies vaccine, which I think was used for many, many years. And it's only recent that we have a vaccine that is better than Louis Pasteur's vaccine against rabies. By the way, vaccination is derived from the word vacca, which is a word that means cow. So vaccination came from Edward Jenner's first vaccine, where the cowpox was used against smallpox. And I guess I should mention the cowpox virus was a virus that hit cows, causing cowpox, but people could get it, and then it protected against smallpox. This protection is called immunity. Any question about any of that? All right, out of all of this work, particularly Louis Pasteur's work and Dr. Koch's work, came the idea that chemotherapy could be used to treat diseases. As well, you probably don't know, chemotherapy, as it was originally used, and when it's used in a broad sense, it means the treatment of disease with chemicals. Now, usually when we use the term chemotherapeutic agent or chemotherapy, we're talking about an anti-cancer drug, okay? Because that's the way we use chemotherapy. However, initially, it was actually used to treat any disease by a chemical. Nowadays, we usually use antimicrobial chemical to discuss this. We don't call it a chemotherapeutic agent to prevent disease. We call it an antimicrobial. Chemotherapeutic agents can be a synthetic drug, which means they're man-made, or they can be an antibiotic, which the textbook defines an antibiotic to be a chemical produced by a living organism that inhibits or kills other microbes. And this is a textbook definition. When you go into the clinic, the general public does not use the term antibiotic that way. and most clinicians do not as well. When we use the term antibiotic, it is any drug that prevents microbes from growing, particularly bacteria, but any microbe. And that's just the way that antibiotics is used in general. But the textbook will define it as a chemical produced specifically by a living organism to prevent the growth of other microbes. So by this textbook definition, penicillin G and penicillin V are antibiotics, but other members of the penicillin family, such as ampicillin, the third antibiotic developed, as well as carboxycillin and all those other cillins that are man-made, they are not antibiotics because a human made them, or at least partially made them. Like ampicillin is actually started as penicillin G, and then somebody takes the penicillin G in the lab and changes the molecule to make ampicillin. Any question about any of that? Surprisingly, antibiotics were not the first chemotherapeutic agent or antimicrobial agent discovered. The first antimicrobial agent that was discovered was quinine from the bark of the cinchona tree. And even today, it is used to treat malaria as long as the malaria is not resistant to the quinine in the bark of the cinchona tree. Now, we don't use the bark of the cinchona tree anymore. We will purify it out as quinine. So I was saying quinine is today the preferred way to treat malaria if the malaria is not resistant to quinine. Now, there are many regions of the world where malaria is resistant to quinine. Paul Ehrlich found the first drug other than quinine to treat a disease. And what he did was he found salvarsan, a synthetic arsenic drug, to treat syphilis. And he did this in 1910. You don't need to know the year, but he proposed that we could find a drug to treat any disease, and he called that drug a magic bullet, a bullet that would kill the microbe killing disease, but leave the patient's cells unharmed. Any question about any of that? So Paul Ehrlich came up with the concept of the magic bullet. However, the first person to come up with a really effective drug to be used against a microbial infection was actually Alexander Fleming, who discovered penicillin. What Alexander Fleming did was he streaked out a plate, and he was a lousy microbiologist. As you can see, he streaked out the plate, and as he was streaking, some microbes fell into his plate. And those microbes were penicillium mold. And he happened to grow the plate. He just left it on the counter so that both the bacteria and the mold grew. And then after the weekend, he came back and found the plate that he streaked out. There we go, streaked out, and realized that he had forgotten to put it in the incubator. And he looked at the plate and saw both microbes, a bacteria, and a mold colony growing on it. Instead of just throwing it away, he looked closely at the plate and observed that around the mold colony, there was no bacterial growth. And he correctly concluded that something was being made by the penicillium mold and was being secreted by them, and it was diluting through the agar so that we had a zone of inhibition around the mold, that prevented the growth of bacteria. And he called this penicillin after the penicillium mold. So one ends in an N, the other ends in an M, meaning the bacteria, the fungi grows in a colony called penicillium with an M. And then the drug that it makes is called penicillin with an N. He was not able to purify penicillin because of the early year he did it in 1928, discovering penicillin. And so penicillin was not used in the clinic until World War II. And before that, we had the first clinical antibiotic that was developed and then became available to use in the clinic. And this was, sulfonamide, a sulfa drug, which was actually a man-made antibiotic. It first became clinically available. Penicillin became the second clinically available drug. And then in the 1940s and actually World War II, they grew up penicillin in mass culture, were able to purify it, and then use it on patients. And of course, it had a great effect, curing bacterial infections. Whoops. Oh, come on. Oh, fudge. Sorry, my mouse isn't working. I cleaned it, so for a while there, it was working much better, but it's not working again. All right. Bacteria were once classified as plants, hence the term flora or microflora. And this is the term that I learned when I took microbiology. It has now been replaced by microbiota. And if you ever hear me use the term flora or microflora, you can remind me of the correction that I shouldn't use microflora because microbes are not plants, for one thing. And then that's the old term. We should call them microbiota. The normal microbiota are microbes that are normally present in and on a person, at least the human normal microbiota, but it's been found that all animals and plants are symbiotic to microbial communities that are actually central to the animal or plant's welfare and to the humans too, meaning microbes do help us in a variety of ways. Come on. The normal microbiota is found on and in the skin, the oral nasal cavities, the respiratory tract, the digestive tract, and the urethra. The lower respiratory tract usually does not have microbes, but the upper respiratory tract is covered with microbes. The normal microbiota is not usually found internal to the body, such as in the blood, the internal organs, or the internal tissues. The normal microbiota are helpful to the host, meaning the people, because they can prevent the growth of pathogens. They can compete for resources. The normal microbiota can secrete toxins that actually inhibit the growth of pathogens, such as E. coli, can prevent the growth, assuming that it's making a molecule and secreting it. E. coli can actually prevent the growth of various pathogens like salmonella and what the heck's the other one? Drawing a blank on that other human disease of the skin, which usually gets internal and then causes more problems, but does cause skin disease. So salmonella is one. Shigella which is mostly an intestinal tract disease, is the other. So E. coli can prevent the growth of these two by producing a toxin that prevents the growth of those microbes. And then our normal microbiota help us obtain nutrients, digest food, and may help us get growth factors. So the normal microbiota or an intestine is largely responsible for helping us digest our food. And for example, the cow eats grass, but a cow cannot by itself digest the grass and get any food value from the grass. What happens is the cow has to have a, I think it's a ciliate in it that has a microbe in it, and it's actually the microbe which digests the cellulose in the grass. Any question about that? For us, E. coli helps us digest our food, the nutrients, and certain growth, factors that we get include folic acid and vitamin K, which we get a significant amount of both of these vitamins from our microbes in our gut, in our intestines. Any question about any of that? All right, the normal microbiota do not normally cause disease. However, they can cause disease in certain circumstances, such as when a patient is immunosuppressed or an AIDS patient. The normal microbiota can cause an infection in them. An infectious disease is one caused by a microbe, and we call a microbe, if it can cause a disease, a pathogen. So all infectious diseases are caused by a microbial pathogenic organism. Any question about any of that? All right, we're going to talk briefly about emerging infectious diseases. And these are new or changing diseases that are increasing or have the potential to increase in incidence. Does anybody know a new emerging infectious disease? Can anyone?
COVID-19.
Yeah, COVID-19. Which has obviously come out and caused disease. There are other diseases, like scientists are watching the bird flu to see if it will ever spread among humans. At the moment, bird flu only spreads among birds, but birds can transmit it to human. And fortunately, the virus, when it infects a human, it does not come out of that human and infect other people, meaning bird flu currently cannot spread person to person. But people are watching it because they're afraid that at some point in the future, it can become an emerging infectious disease. So it has the potential to increase in incidence. If you don't know, bird flu is highly toxic, not toxic, highly lethal. And I don't remember the percentage, but it's more than 10% of the population that come down with the bird flu will die of the bird flu. And I think it's less than 50%. So it's someplace between 10% and 50%. And I don't remember exactly how much, but it's much greater than COVID-19, for example, the lethality of bird flu. And that's it for emerging infectious diseases. All you need to know is the definition of what an emerging infectious disease is. Any question about any of that? If not, let's move to Chapter 2, because we were supposed to start this today and I'm behind schedule. Let me shut down the first one. If I can get the mouse to work. Come on. There we go. I think I changed the two here. So there we go. Yep. All right. So in the first slide, I show you the major goals and a rough outline. Know the terms of this lesson. What's an organic and inorganic compound, hydrocarbons, polar and nonpolar molecules, what's a hydrogen bond, what's an acid, what's a base, and the pH. I also am going to give you a rough outline. We are going to talk about different things with respect to the 4 major biological molecules, be able to describe the 4 biological molecules, their relationship and characteristics of the monomers and the polymers, be able to describe the composition and structure and function of the 4 major biological molecules, and then be able to give examples of each. For the 4 major biological molecules, you can find them on almost any item of food, with the exception of nucleic acids. 3 of them are listed, and they are carbohydrates, lipids that we call fats, and the third being amino acids and proteins. And then the fourth being nucleic acid. And you will not find nucleic acid on any box of cereal or any other box that I'm aware of. Generally, in processed food, you wouldn't find much nucleic acid anyways. But if you're eating raw fruits and vegetables or anything else that's raw, you will be consuming nucleic acid. And did I say be able to give examples of each of the 4 biological molecules? We're going to talk about the scale of things. You do not need to memorize this slide. Let's see if I can blow this up. Here we go. But you should know the relative order. One of the largest things we're going to talk about in this class are people. And smaller than people are the chicken egg. Smaller than chicken eggs are frog eggs. And frog eggs are about the smallest thing our human eyes can see. We can see a little bit smaller than frog eggs, but not much. And then below that, we have the size of most eukaryotic cells. A typical small eukaryotic cell would be a red blood cell. And then all other cells of the body would be other eukaryotic cells, which most of them are larger than the red blood cell. Below that are bacteria, and they're mostly between 1 and 2 micrometers in size, or about 10 times the size of eukaryotic cells, or 10 times smaller in size. So eukaryotes are 10 times larger, about, than a prokaryote. What's smaller than bacteria is viruses. Instead of being a cell, they usually only have a protein coat and then an internal molecule, the nucleic acid for their genome. And some viruses have a lipid envelope around the protein coat. Smaller than a virus are large molecules like proteins. Smaller than a protein are small molecules like water. And then smaller than small molecules are atoms. Any question about any of that? Like I said, you do not need to memorize this slide, but you should know the relative order of the different objects on the slide. And let me point out that there is a mistake in the slide right here. It's got micrometer right here, 1 micrometer. Come on, mouse. My mouse isn't working. There we go. 1 micrometer right here. And then there's 100 micrometers, which is supposed to be smaller than that. That's a mistake. That should be 1 millimeter. 1 micrometer is actually down here. Any question about any of that? All right. So what are organic compounds? Organic compounds are molecules that typically bind to other molecules, and they typically have carbon holding the organic molecule together. Typically, organic compounds are complex compared to inorganic compounds. Most of the bonds in an organic compound are held together by a covalent chemical bond. An inorganic compound typically lacks carbon, but not all of them lack carbon. Inorganic compounds are required by living organisms for growth, repair, maintenance, and reproduction. Some examples of an inorganic compound that are very important to life include oxygen, carbon dioxide, as well as water. These are all inorganic molecules. And water, for example, is the main molecule in everything alive. But water is not an organic compound, but it is one of the most important molecules in the body. Any question about any of that? Oops. All right. What is a polar molecule? A polar molecule is a molecule that has the electrons not shared evenly between the atoms of that molecule. Water is a polar molecule, and the electrons spend more time with oxygen than the electrons spend with hydrogen. However, the electrons do not move from hydrogen to oxygen. They just spend more time being near and or going around oxygen than they do hydrogen. And this is a polar molecule, not a complete charge, meaning it's a partial charge. And your textbook does not use the term partial charge. I don't remember what the textbook uses, something like incomplete charge. Because if it was a full charge, around the oxygen, we'd have that a -1, and around the hydrogen, we have a +1. And what my point is, this is a partial charge, not a complete charge. Water's polarity allows water molecules to bind to 4 other water molecules. So for example, here we have a water molecule in the center. The partially positive hydrogen will be attracted to the partially negative oxygen of this molecule, forming a hydrogen bond between them. And then this hydrogen over here can form a hydrogen bond to this oxygen and the water molecule over here. And then we have 2 other water molecules where the hydrogen can form a hydrogen bond to the oxygen here, meaning the partial hydrogen, this partial +1, partial, not complete, is attracted to the partial negative charge on the oxygen. Any question about any of that? If not, let's watch a little video. There we have 2 hydrogen atoms forming hydrogen gas. And there's an oxygen molecule forming oxygen gas. And there's 2 water molecules having a oxygen that's blue and a hydrogen that's green, or in this case, red. there's a polar charge, a partial positive on this hydrogen, partial positive on that hydrogen, and then a negative charge, partial negative charge on the oxygen. That means this oxygen will be attracted to the partial hydrogen of another molecule, creating the hydrogen bond between them. You'll note that at the temperature of liquid water, the hydrogen bonds break and reform all the time. And that's why the liquid in this cup takes on the shape of the cup. The hydrogen bond that breaks and reforms all the time goes around each other. And let me block that there. Doesn't form a permanent bond. Hydrogen bonds are very weak, and at the temperature of liquid water, there's enough energy to break that hydrogen bond, at least some of the time, not all of the time. And it depends on how much cosmic radiation you're getting and a few other factors. Any question about any of that? If not, we're at a good stopping point. So I'm going to stop. Well, wait a minute. This is the summer. Let me check when we're supposed to stop, because we go longer in the summer. Yep, got to go to the top. $6.50. See, I was thinking 6:15, which is the normal term when we stop, but we still have to go to 6:50. So let me continue. And then we will not be so far behind in chapter 2. The water is a polar molecule. Water is also a good solvent. All polar molecules can dissolve in water, as well as all ions can dissolve in water. A polar molecule is one like water, where the molecule has a partial positive on one end of the molecule and a partial negative on the other end of the molecule. And so we call water a polar molecule. Any molecule that has this arrangement is called a polar molecule. For example, if you were to put glucose in water, and then you would get sugar water, you would have hydrogen bonds forming between the sugar of the water, as well as the hydrogen of the sugar would bind to the hydrogen, not the hydrogen, the hydrogen of the sugar would bind to the oxygen of the water. So polar and ions can dissolve in water. How that happens with a ion is the water comes up to the ion like an ion crystal, and then removes the molecule from the crystal, and then surrounds the negatively charged, because sodium chloride has a full -1 charge. Water will then surround the chlorine atom with the hydrogen atoms being focusing and pointing towards the chloride ion, so that the partial charge of water will be next to the negative charge of chlorine. And to equal out that negative charge, you need to have several molecules binding to the chloride, which is usually not a problem, because in our blood we have lots of water, and it doesn't matter what the water came from. It can come up to the hydrogen and then pull it out. Any question about any of that? All right, polar substances dissociate in water. Like if we put a salt crystal in distilled water, the water will start dissolving the salt as the sodium ion, the sodium ion and the chloride ion dissolve into the water. Any question about any of that? All right, let's see if this still works. If it doesn't work, I'm not going to show it if it doesn't work, but let's see if we can get it to work. It used to work. The question is, does it still work? And I don't know the answer to that. Looks good. This might be the same thing we saw. Nope. Water dissolves in salt. A water molecule. A salt crystal. Water comes in. It's attracted to the sodium chloride. The sodium chloride forms a hydrogen bond to the water. Several water molecules come in and pull off the ion of either sodium or chloride. So these two are attracted to the oxygen. And then these, let me go back on that. So here's water coming in around the sodium, surrounding it by putting the negative portion of oxygen in water towards the positive charge of the chloride ion. Is that right? Water dissolving the salt, pulling away the ions. Sodium has a different orientation to the water molecule than chloride, because sodium has a +1 and chloride has a -1 charge. Any question about any of that? What did I do? It's probably here. There it is. I think that's my mouse joining and leaving the computer. So water is a good solvent in that it can dissolve any polar molecule. Polar molecules are hydrophilic. That literally means lover of water, hydrophilic. But what we mean by that is not that a hydrophilic molecule actually loves the water molecule. It's just they're attracted to each other. For example, sodium chloride is attracted to water and then will form salt water as it's dissolved. And the point is that sodium chloride is either polar or ionic, and that means it's hydrophilic, that it will dissolve in water. Any question about any of that? For an example of nonpolar molecules, these are molecules that are repelled by water, and we call those hydrophobic molecules. Literally, hydrophobic means fear of water, water, phobic, fear of. But what we mean by that is a hydrophobic molecule is one that will not dissolve in water. I'm sure all of you have made salad dressing And when you pour in the oil and the vinegar and the water and all your seasoning, and then you shake it up, the oil will quickly separate and then form a layer on top. Any question about any of that? Hydrogen bonding between water molecules gives water many of its properties, including making water excellent at buffering the temperature. You guys are mostly from Western Washington, maybe a few from Western Oregon. As you know, the western parts of our state have their temperature buffered more than in Eastern Washington and Eastern Oregon. So Eastern Washington and Eastern Oregon, in the summer, it's much hotter than it is in the central and coastal regions of the two states. And that's because of water buffering the temperature. The water from the ocean buffers Western Oregon and Western Washington, so that in the summer, our temperature is less hot than the east side of the state. And then in the winter, the water also buffers our temperature, and our temperature is much milder, meaning less cold, than somebody in the east part of the state. Any questions about any of that? Why water is excellent at buffering the temperature is that when we apply heat to water, there are two ways that energy from the heat can be absorbed by the water. First, the water can absorb energy into the hydrogen bonds, breaking the hydrogen bonds between the water molecules. And therefore, if, then the water evaporates. So therefore, the water molecules that are evaporating are the water molecules having the most energy. And if a water molecule has the most energy, that water molecule has the highest temperature. Because temperature, all it is actually a measure of how much energy a molecule has. And it's actually the measure of how much the molecule is moving around. So jumping around or moving around. That's one way energy can be absorbed by water. It breaks the hydrogen bonds, and then the hottest water molecule in the water leaves. That leaves the rest of the water relatively cool. And as you know, that is why humans perspire, because water is efficient at cooling us down when it perspires, and then the hottest water molecule will be the ones leaving our body, and that will help cool us. Any question about any of that? All right, the second way a water molecule can absorb energy when we apply energy to the water is that the water molecules themselves can absorb the energy and then move around faster. What you do is, when you do that, is you're increasing the temperature of that water molecule. And remember, the temperature is just a measure of how much a molecule is moving. Water has two forces or two ways it can absorb the energy we're putting in as heat. It can absorb the energy by the hydrogen bonds absorbing it, and it can absorb the energy by the molecules themselves. Most other substances like oil, concrete, whatever else you can think of, unless it has water in it, most other substances only have one thing that can absorb the heat, and that is the molecule itself. And therefore, if you give the same amount of energy to a container, let's say 100 mils of water into 100 mils of oil, that same amount of energy, like 100 joules, will increase the temperature of the oil more than it will increase the temperature of the water. And that's because some of that energy going into the water is going to be absorbed by the hydrogen bonds. And that molecule will then leave the water and evaporate, taking a lot of energy with it when that water molecule leaves. Most other substances only have one thing, like the oil, only the molecules can absorb the energy. And so oil will rise higher with the same amount of energy compared to water. Any questions about any of that? All right. The next thing I need to talk about is water can split into H plus ions and OH minus ions on the temperature on the surface of the planet Earth. Okay. Now in the sun, let me go here, come on. In the sun, the temperature is hot enough you can break water down into the atoms. That does not happen on the surface of the planet Earth. So water, which is H2O, and I can probably even come on, my mouse now died again. There we go. Make that a subscript. Up, darn it. And then blow this up. Make it bigger so you guys can see it better. So water can split on the planet Earth into H plus ions plus OH minus ions. Superscript that now. Superscript that. On the planet Earth, at the temperature of the normal Earth, water does not split into H plus ions, meaning two of them, and O minus two ions. That would have to happen at the surface of the sun, which is much hotter than the surface of the Earth. And it is true on the planet Earth, you can split water. So let's go ahead and do that. Oops, come on a little further. You can split it into two H pluses and OH minus 2 on the sun, or in electrolysis, meaning if you put energy into here, I'm not sure how to put that in there, so let's just go here. And get that out of there. You put electrons in there in electrolysis. You can also do this. But this does not normally happen to water. You have to pump in electricity into it to split water into H plus ions and O minus 2 ions. Normally water by itself only breaks into H plus ions and OH minus ions on the surface of the planet Earth. Okay, any questions about any of that? So if you get asked this question and you put this, I'm letting you know this is going to be wrong. This doesn't happen on the surface of the planet Earth, at least normally. This is important for giving water some of its properties. The charged molecules, meaning the H plus ion and the OH minus ions, can participate in chemical reactions and give the water properties. The main property it gives water is it gives water its pH. Let's talk a little bit about pH. Here we're looking at the pH scale. Let me blow this up. Move that up. So there it tells you it's the pH scale, which the very top is 0, which isn't hidden here. And neutral water, when water first breaks apart from H2O into H plus and OH minus, will be pH 7, where the concentration of H plus ions equals the concentration of the OH minus ion. So that'll be pH 7, which is pure water. And human blood and milk is very close to pH 7. I think blood is like pH 7.3 to 7.4, and milk is slightly acidic, but very close to pH 7. Only neutral pH 7 is where H plus ions and OH minus ions equal each other. When the pH is lower than pH 7, we're in an acid environment where the concentration of H plus ions is greater than the concentration of OH minus ions in solution. So urine around pH 6, slightly acidic. Wine and tomato juice, more acidic. They're pH 4. Grapefruit juice, pH 3. Lemon juice, pH 2. And then stomach acid being about pH 1.5. To get around pH 1, the only thing I know that's around pH 1 is the acid in the battery of our cars. That's the most acidic thing that I know of. I don't know something that is more acidic, but it's possible that there's something that is. As you go down the scale, meaning the pH lowers from pH 7, getting lower down to 0, the environment becomes more acidic as the H plus ions increase in solution and the OH ions decrease in solution. When you go up in pH, you become a basic solution. And as you go up from pH 7 to pH 14, it becomes increasingly more basic. So seawater is slightly above pH 8, slightly basic. Milk of magnesia between pH 10 and 5, more basic. Household ammonia, more basic, around pH 11.5. Household bleach is around pH 12.5. And then oven cleaner is the most basic thing you guys are probably familiar with. It's above pH 13, about pH 13.5. If you've ever heard of lime water, it approaches pH 14. So the pH scale ranges from pH 0 to pH 14. At pH 0, all of the water molecules are broken down into H plus, and then all of the OH molecules are gone because there's none in solution at pH 0. At pH 14, once again, all of the water molecules are broken down into H plus and OH minus, and all of the OH minuses are present at pH 14, and all of the H plus ions are missing at pH 14. Any question about any of that? All right. Let's go on in the show. So an acid is a substance that dissociates in solution, giving off one or more H plus ions. HCl is an acid because when it's dissolved in water, the HCl will break apart into H plus ions and chloride minus ions. And there's more H plus in solution, so it's an acid. Any question about that? Now your textbook says a base is a substance that dissociates and gives off OH minus. So sodium hydroxide is a base because when you dissolve that in water, it'll break apart into sodium ions or Na plus ions and OH minus ions. So sodium hydroxide, NaOH, sodium hydroxide is a base because it will release OH minus. Your textbook does not tell you this, but I am telling you this, that a base is also a substance that when it dissolves in water, absorbs H plus. So a base is something that either gives off OH or something that absorbs H plus. For example, bicarbonate, HCO3, can absorb H plus and then form carbonic acid, H2CO3, and that will lower the H plus concentration in solution, making the solution more basic. And if you don't know, sodium bicarbonate is baking soda is a base when you add it to water. And this is why, because it absorbs H plus ions. So a base is both something that absorbs H plus ions or something that absorbs, releases OH minus ions. And your textbook actually got that slightly wrong. It only gave one possibility for a base, and there are two possibilities. Any question about any of that? All right, let's talk a little bit more about things dissolving in water. A salt is a substance that dissociates in waters into a cation, something that has a positive charge, and an anion, something that has a negative charge, neither of which are H plus ions or OH minus ions. For example, sodium chloride, salt, dissolved in water, will dissolve into Na plus, a cation, and chloride minus ion, an anion. So sodium chloride, which everybody calls salt, is actually a salt. However, chemists will tell you that magnesium chloride is also a salt because it will dissolve in water, and the magnesium forms a cation, and then the chloride forms an anion. So magnesium chloride is, at least for this lesson and for chemistry, also a salt. Anything that dissociates into cations and anions is a salt, besides sodium chloride, which everybody calls table salt. An electrolyte is simply a substance that dissolves in water that is a salt, and then the ions can carry the electrons of electricity. So an electrolyte is something that can carry the charge of electricity, meaning can carry the electrons. And that is why whenever a lightning storm hits, you should get out of the ocean water because it's salt water, and the salt water will carry the charge of the lightning very readily. And if it hits anywhere near you or anywhere even fairly just close, meaning not close at all, you will get a shock from the electricity because salt water carries electricity so well. Whereas if you're in a pool of fresh water does not carry the charge of the lightning very well, although you probably should get out during a lightning storm, but you don't really have to get out because the lightning will have to hit within a few feet of you before you would get the effects of the electricity from the lightning charge. Okay, if it hits the other side of the pool, you'd be safe. You have to be hit with the lightning within a few feet of you. Any question about that? All right, let's talk a little bit more about the pH. The pH is actually the amount of hydrogen ion in solution, and mathematically, the pH is equal to the negative log of the concentration of hydrogen ion in solution. Let me get my web page out again. A pH of 7.0 exactly equals 1 water molecule. Let's just copy and paste. One water molecule breaking into H plus and OH minus. Let me get out of that. Per 1 times, where's my times? 10 to the 7th water molecules. So if the pH is equal to 7, that means that there's 1 hydrogen ion in solution for 1 times 10 to the 7th water molecules. You got that? For a pH of 6, there was 1 water molecule breaking apart to give 1 hydrogen ions in solution for 1 times 10 to the 6th water molecules. You got that? For pH 5, it's the same. Can I glue that again? Yep. 1 water molecule breaks apart for 1 times 10 to the 5th water molecules in solution. And then the same for a pH of 8, meaning go up in the scale. A pH of 8 means there's 1 hydrogen ion present in 1 times 10 to the 8th water molecules present in solution. So that's the meaning of pH. Any questions about any of that? Let me state that most organisms grow best between pH 6.5 and 7.5, meaning they grow best around pH 7. There's most organisms, not all organisms grow best at that pH, but most organisms grow best around pH 7. And that's actually true for humans too. We do best around pH 7. Any questions about any of this? If not, I'm going to end it here, and I will see you in the lab at the regular lab time, which is, I think, 7 o'clock. All right, if there's no questions, I'm going to say goodbye.
I did have a question about the extra credit assignment to get together with students for study groups. There's no in Canvas, there's no like way to like chat. There's no chat thing
There's
in
no
there.
chat, but you can email them, and that's a better way to contact them. I can maybe create a discussion board, and then you guys can talk on the discussion board, but you'll have to remind me in the lab to do that, and I'll try and do that.
Okay, that would be great.
Okay.
Thank you.
All right.
Mhm.